Welcome to Pharmamar. Pharmamar is an integrated biopharmaceutical company, and here we undertake all the activities of drug discovery, starting with the marine organisms, developing a synthesis, formulating the compounds, and then clinical trials and hopefully commercialization. And today we're going to visit the R&D laboratories and see how we do this process. And you'll be able to see through my eyes how we do each different step of the process. So please follow me and we'll start the visit. Welcome to the Expeditions Department, the start of our drug discovery process. Please, follow me. The drug discovery process begins with the diving expeditions to collect samples of the marine organisms. Pharmamar employs a team of marine biologists and professional divers who spend most of the year at different locations around the world collecting samples. The samples are collected by hand by scuba diving with special breathing equipment that allows us to dive to below 120 meters. Just a few grams of each organism is sufficient, and so there is no impact on the natural environment and the biodiversity in the sea upon which we depend. It's very important that the samples are frozen at the time of collection to prevent degradation during shipment back to Madrid. All the relevant data about the origin and appearance of each sample is recorded, including an underwater photograph, to help us classify the organisms and to understand their taxonomy. When the organisms arrive at Pharmamar, the IT system assigns a code, a NORMA code, and this code allows us, with a barcode, to access all the data about the samples. The database is called Nautilus, and here we can see an example, in this case a sea star that was collected in the South Pacific, and the database provides all the information about the sample, the GPS coordinates, the depth of collection, and information about the taxonomy of the sample. Over the years, Pharmamar has amassed an enormous number of different organisms, and today the collection is more than 150,000 different marine organisms. To start the discovery process, we first generate a crude extract of each organism. And here we try to extract everything possible from the sample. The next step is to prepare a plate for screening to see if any of the different components show activity against cancer cell lines. And then we know we have an active component and we can continue the process of purification. Now we're going to look at what we do with the marine microorganisms. The first step in the process is to isolate and characterize the thousands of different bacteria that we find in the marine environment. We can also test the different bacteria to determine their genetic composition and their molecular taxonomy. This allows us to make sure that we're working on novel organisms, and we can avoid working on repeat organisms that we've studied in the past. Each different microorganism is cultivated and a crude sample prepared to test in vitro to see if any of the metabolites have activity against cancer cells. The most interesting bacteria that produce active metabolites can be scaled up, first into reaction flasks and then into larger fermenters. And that way we can take larger quantities of the active metabolites for further research and to develop the compounds for clinical trials. Now we can see how we test the different samples for their activity in vitro. Let's have a look inside the cell culture laboratory. So this is where we store and work with the different cell cultures we use in the in vitro testing of the samples, both from macroorganisms and from microorganisms. The in vitro screening process is conducted using a 96-well plate like this one. Each well contains cancer cells and a sample to be tested. We use 4 different cell lines in the primary screen: breast, lung, colon and pancreas. The test produces a changing colour. The active ones, the ones that are interesting to us, are then further purified so we can isolate the pure compound responsible for the activity. Each year we process thousands of samples: crude extracts, fractions from the purification, and finally pure compounds. For the pure compounds, we test these in a larger panel of cells to get an idea about the tissue selectivity and the possible mechanism of action of the compounds. And these are both important aspects for selecting compounds for further development. Now we've arrived at the natural products chemistry group. And this is where we purify the crude extracts and isolate the new pure compounds for further testing. So the first part of the purification process involves passing the crude extract from the marine organism, be it a macroorganism or from the microorganisms as well, through a solid support. And this generates fractions. The fractions are purer than the original crude extract, and in this way we gradually purify the sample. The final part of the purification process is conducted by high-performance liquid chromatography, or HPLC. This guarantees that we obtain a very pure sample of each of the new compounds that we isolate. The quantity is only very small, just a few milligrams, barely enough to see, but sufficient for us to be able to work out the chemical structure. Okay, let's have a look now how we're able to work out the chemical structures of the new compounds we find in the marine organisms. The chemical structure of new compounds is elucidated using powerful nuclear magnetic resonance machines. The sample of interest is introduced into the middle of a powerful magnetic field, and this generates a spectra with detailed information about the hydrogen and the carbon atoms in the molecules. The marine environment provides us with very complicated and diverse chemical structures. But even so, with a few milligrams, we can normally work out the entire chemical structure of each new compound. Over the years, we've isolated many new compounds, and this attests to the great power of the marine environment as a source of new chemical entities to fight cancer. As we have seen, nature only gives us a very small amount of the new compounds. So to be able to continue the work, we need to develop a solution to the supply problem. That's typically done by chemical synthesis. And that involves building up the complicated structure of the natural molecule using small building blocks that we can buy commercially. This can typically be a very long process because the molecules are very complicated. But so far we've been very successful, and all the compounds in clinical development are currently produced by synthetic processes developed by Pharmamar. Here we can see how once we've developed a chemical synthesis that works on a laboratory scale, we can start to scale up that process and get larger quantities of the intermediates and ultimately of the final active drug substance, which allows us to do more detailed analytical studies and to start to think about a formulation for animal studies and ultimately for use in patients. Non-clinical experiments are an essential part of the development process. The first studies, called proof of concept studies, are designed to demonstrate in vivo activity in several different tumour types. This confirms the original activity that we saw in the in vitro testing. Here in the bioanalytical laboratory, we test samples in biological matrices, such as plasma, urine, and other tissues, including tumour tissues, obtained from the animal studies. This allows us to understand how the different compounds are behaving in an in vivo system, and tells us about the metabolism, and gives us clues as to how to best administer the compounds to get the best possible efficacy and to limit the toxicity as much as we can. Once we have larger quantities of the compounds that are active, we need to think about preparing a formulation. For patients, the preferred formulation is a freeze-dried vial that is administered intravenously. To prepare these vials, an aqueous solution is prepared of the compound, and this is accurately dosed to vials so that each vial contains exactly the same quantity of the active substance. The vials are then introduced into a freeze dryer and the aqueous solution is frozen. Then, by controlling the temperature and the pressure, we can slowly evaporate the water, leaving a solid cake that is the final format. The drug substance in this format is stable and can be stored and transported.